Okay, so the structure and organization of bacteria chromosomes. Alright, so here we're looking at a quote-unquote typical bacteria chromosome. They are round for most species. There are a couple that do contain linear chromosomes. You'll notice also that most bacteria species have a single type of chromosome, so just one chromosome, same sequence. But they can have 1 to 4, depending on the conditions and depending on the species. Okay? A chromosome is a few million base pairs in length, and that has about several thousand genes interspersed. So this represents a gene, and then we have short intergenic regions that separate them. And then we also have some repetitive sequences in our bacterial chromosome. There's one origin of replication, as we talked about before. Okay? And the purpose of these repetitive sequences is to assist in DNA folding, gene regulation, and genetic recombination. So typically circular, typically one type per cell. So here we have a bacteria cell, and all of its DNA is outside of the cell. So what process can occur now? I hope you remember. Now look at all this DNA, and look at this cell. All of that DNA has to fit inside. And this is the reason we have to compact our DNA. This has to come in 1,000-fold to be able to get all of that DNA inside of a single region within the cell and still have room for other organelles. So there's ribosomes in here. I'm trying to think what else might they have. And other little pieces, not like what's in an animal cell, but they do have some things within them. So here we go. So this is an artist rendition. You see how much you have to compact it. So you have to go from everything you see here to fit nicely inside the nucleoid region, and then have other stuff that has to go inside of this cell. Now if we look at it in real time, remember, they can have 1 to 4 DNA molecules depending on the species. So here they have 2, and you'll notice that each nucleoid contains a chromosome. So there's 1 chromosome per nucleoid. So if they had 4, they would have 4 regions that have these chromosomes in them, so they are all separate. Now, how do we get it? So how do we go from all of this to that, to packaging it inside the nucleoid region? So we start with the circular chromosome, and now we have to form loops. These loops are called microdomains in bacterial chromosomes, and each microdomain is about 10,000 base pairs. Then, if you put 80 to 100 microdomains together, you get a macrodomain. And now these loops are maintained by these nucleoid-associated proteins, okay, also called NAPs. Now, they can bend the DNA, they can act like bridges that cause DNA to bind, they can facilitate chromosome segregation, they can play a role in gene regulation. So some examples of NAPs are these histone-like nucleoid structuring, so HNSs, and structural maintenance of chromosome, SMCs, which are also found in eukaryotes. So what does that tell you? If you have a protein in bacteria and eukaryotes that have a similar job, think about that. So then, after we get the macro and micro domains, then we're going to introduce this supercoiling. So imagine taking a rubber band and twisting it between your fingers and how it folds onto itself. That's similar to what happens here with this supercoiling. So bacterial chromosomes have this negative supercoiling. And there are 2 enzymes within the bacteria, so DNA gyrase and topoisomerase. And their job is to introduce supercoils and relax them. So DNA gyrase introduces negative supercoils and relaxes positive. Topoisomerase introduces positive and relaxes negative. So what does that mean? It's the way in which they're folded. Okay, so you'll see negative versus positive. And it's specific to what the chromosomes are. And now it has a purpose. So, of course, we know that this negative supercoiling allows compaction. So we do this to reduce our DNA 1,000-fold. But in relieving these supercoils, it also allows for strand separation. Now, why is that important? So think about it. If we have to access this DNA, it's easier to access it when the strands have separated. So this will allow proteins to get inside when it's time for replication and transcription and any other reason that you would need to get inside the DNA molecule. Interesting enough, this also poses assistance for designing pharmaceuticals, so antibiotics. We can target the bacteria topoisomerase molecules, and they're different than eukaryotes, so if we target these, we can stop the bacteria from being able to grow. If it can't introduce and relax supercoils, it can't function because it can't compact this DNA and it can't have strand separation, and without strand separation, it can't transcribe, okay, or replicate. So you should be able to take a few minutes and think about what you know about the DNA in prokaryotes, and you can fill in that on your chart now. Okay? And if you have any questions, comments, or concerns, you know what to do.